students, so under animal kingdom we have completed with the 6 bases of classification which Whittaker used for classifying the animals into the 11 phyla. In NCERT there is a flow chart. These are the 11 phyla and they have been classified accordingly in this manner. Kingdom Animalia is divided broadly into 2. In cellular level organization there is just one phylum, that is Phylum Porifera, and you see that the symmetry is mostly asymmetric in poriferans. Body cavity or coelom is absent. They are acoelomates. Only one phylum in that category, that is Phylum Porifera. All the other phyla are classified in the second group, that is tissue, organ or organ system level organization. Again broadly divided into 2. There are some phyla that have a radial symmetry and the others have a bilateral symmetry. Having a radial symmetry is a less evolved feature of animals and bilateral symmetry is a more advanced feature. Under radial symmetry there are 2 phyla here, Coelenterata and Ctenophora, and they both are acoelomates, diploblastic animals, so there is no coelom involved here. In bilateral symmetry, there is this phyla Platyhelminths, which is a triploblastic phylum if you remember. However, despite being triploblastic, it does not have a coelom. They are acoelomates. The phylum after Platyhelminths, that is Aschelminths, which is also triploblastic. In this particular phylum, the mesoderm is trying to produce coelom. However, the coelom may not be properly formed. They are formed in the form of pouches like structures. That kind of a coelom is called a pseudocoelom, which is found in the Aschelminths. Okay, they are not called acoelomates, they are called pseudocoelomates. It is from Annelida onwards, they are all triploblastic and coelomate animals, wherein the mesoderm of these animals has split into a proper functional coelom. So all these phyla, remaining phyla will be coelomates. How many phyla are there in under coelomates? 1, 2, 3, 4, 5, 6. 6 phyla are placed under coelomates. So this is the flow chart that is present in NCERT. What we are going to do now is we are going to study each of this phylum in great detail. Okay, shall we begin? Let's start with the first phylum, that is Phylum Porifera. Let's begin with the first phylum, that is the Phylum Porifera. Porifera means pore-bearing animals, also called as sponges. You must have seen a sponge, how it has holes all over it. Even the sponges, these are animals bearing pores all over their body. They are found in aquatic habitats only, mostly marine. Some few freshwater forms are also present. They may be present singly also, or they may be colonial. They are mostly sessile. Sessile meaning they are not motile forms. They do not move around except in the larval stage. They are animals, okay, stationary at one place throughout their life. If you look at their body, it will look like a vase-like structure with pores all over its body and a peculiar bigger hole on top. Okay, every sponge will have this kind of a hole on top. Or what it does is it will just absorb the water through all these pores on its body, and there is a cavity inside. The water that flows inside through these pores, okay, enters into a cavity inside the body, wherein the animal will uptake food from the water, oxygen from the water, and eliminate its excretory products, etc. The water that come in may also carry gametes from some other sponge, and fertilization may also happen right there, and then the wastewater is then just expelled out to the pore on top. This is the pattern that they follow, a very primitive kind of a pattern. Habitat, they are aquatic, mostly marine, a few freshwater forms. Example, Spongilla, a popular example of a freshwater form. Habit, they are sessile. Sessile means sedentary at one place. Larval stage is motile. What is the level of organization? Is cellular level of organization. Symmetry, most forms are asymmetric and very few forms are radial symmetry. Germ layers, are there 2 or 3 germ layers? 2 germ layers only. They are diploblastic. However, because they do not have a tissue level organization, some textbooks may say that the term diploblastic is not applicable for poriferans. The term diploblastic can be applied for Cnidaria onwards. In NCERT, for diploblastic organization, coelentrates has been mentioned. Germ layers, we can say diploblastic since they have 2 germ layers, but you can just mention over here, not diploblastic in true sense. since they have cellular level organization. See, if there is a question, who among the following has diploblastic organization, and in the options you have, let's say Porifera, you have Cnidaria, you have Platyhelminths and Esculminths, then you will not go for Porifera, you have to go for Cnidaria. Understood? Coelom, acoelomate animals. Next we talk about the body structure. So they have a vase-like appearance, like I told you. A peculiar opening is there on the outside and many pores are there all over the body. These pores all over the body are called as ostia, and there are many ostia, and this one peculiar opening on top is called as the osculum, acts as the exit, and this act as the entry of water. So I've taken a longitudinal section of this, and as you can see, these are the ostia here, and a cavity is there inside. This cavity inside is called as the spongocoel, a primitive cavity, also called as paragastric cavity, which will act like its stomach. If you have to note the movement of water, it's going to enter from all the sides, all these ostia. It will enter into this spongocoel and it will be released out through the osculum. The spongocoel will act like a primitive gut. Paragastric cavity. Why has it been named after gastric? Gastro means what? Stomach. It's like a primitive stomach. And this right here is the body wall, which we're going to talk about now. This is its body wall. This body wall will be made up of 3 layers. The 3 layers will be formed by the ectoderm. This outer layer will be formed by the ectoderm. The inner layer will be formed by the endoderm, and the middle layer, if you remember, the mesohyl layer will be present in between these 2 layers. Since it is diploblastic, only 2 layers will be properly differentiated. The second layer, middle layer, will not be differentiated. We'll be studying that in more detail. This cavity that you see over here, this is not a true cavity. For a cavity to be a true cavity, it has to be formed by the mesoderm, which is not differentiated over here. It is lying right here, and it is in the form of mesohyl layer, not even a mesoderm. If the mesoderm forms the cavity in between like this, splitting, then it becomes a true cavity, which is seen in us. This cavity that is formed, it's like the gut. That is not a true cavity. Not a true cavity, since not formed by mesoderm. Let us look at the body wall in more detail. Let us zoom out this particular portion right here. What do you see? These pores that you can see across here, these are the ostia. If you remember, the poriferans are diploblastic animals. They have the ectoderm and the endoderm, differentiated. This is the ectoderm that has been differentiated, forming this outer layer. Alright, this outer layer is known as the pinacoderm, made up of these flattened elongated cells, and the inner layer, these flagellated cells which line the spongocoel, this inner layer is known as choanoderm. The pinacoderm is formed by the ectoderm, the choanoderm is formed by the endoderm, and what you have in between is a jelly-like substance, an undifferentiated mesoderm, right, which is not called mesoderm, it's called mesohyl layer. This layer in between, the jelly-like substance, is called as the mesohyl layer. Let's talk about each of these layers in more detail. The pinacoderm. The pinacoderm is made up of flattened cells. The flattened cells are called as pinacocytes, and the function of this pinacoderm is protection. Origin is the ectoderm. Next we talk about the choanoderm. Origin is the endoderm. The individual cells are called as choanocytes. These are said to be flagellated cells. If you look at the individual cell, these are actually the cellular processes. They help in formation of the water current. They help it in trapping the food particles and help it in the process of feeding. This is a flagella, and this is said to be its collar. Collar is made up of the extensions of the cells only. This is a choanocyte, and we can say that the function of the choanocyte is formation of water current, ingestion by phagocytosis, filter feeding, selectively feeding what it wants. That is what it helps in, choanocytes. Then third one, the layer in between is a jelly-like substance which will be called as the mesohyl layer. The mesohyl layer will have several constituents inside it. We're going to study just 3 constituents of it. This is the first one. Amoeba-like cells will be found. These These amoeba-like cells which are found are called as amoebocytes. Amoebocytes are very totipotent kind of cells. They can turn from one cell to the other. They can form gametes. They are mainly meant for transportation, picking up of material or nutrients from one place and transporting them elsewhere. They are amoeboid cells. They are meant for transportation, formation of other cell types. What are these green things? These are called as spicules. What are spicules? So spicules are deposits of certain minerals, okay, elongated deposits of certain minerals like calcium carbonate or silica. They can be calcareous or siliceous. They act as the endoskeleton of sponges and gives them rigidity. Spicules. Spicules are said to be endoskeleton and they are mineral deposits. They can be either calcareous or siliceous. The classification of sponges is mainly based on the types of spicules that are present in the sponges. Third thing that you will find in the mesohyl layer are these. What are these? Such kind of fibers will be found. These fibers are known as spongin fibers. These will be proteinaceous in nature and they also act as the skeleton. Okay, they help to give it rigidity, provide rigidity. Just like how collagen and elastin fibers provide us rigidity, similarly spongin fibers which are proteinaceous give them rigidity. So mesohyl layer, you have to say 1, 2 and 3 things we have seen. Mesohyl layer comprises of amoebocytes, spicules and spongin fibers, and that is the body wall. Mesohyl layer can be also called as the mesenchyme. Choanocytes can also be called as the collar cells because of the collar.
महादेव के नाम पे सब्सक्राइब कर दें।